Hi and welcome to week 2 of our revision course for 5th years for biology. We had a vote this week and genetic engineering was one of the top choices, so I said we'd take a quick look at that. In general, I think this today will probably take us maybe 10 to 15 minutes. What we'll do is we'll go through the key areas involved, and then for each week what I'll do is I'll upload the PowerPoint and some questions for you to try. So if we take a look firstly at it, some of the key things we need to know. Firstly anyway, genetic engineering is the artificial alteration of a gene. Essentially, this involves cutting a small section of DNA from one organism and inserting it into the DNA of a second organism. So essentially what you're trying to do is you're trying to include one characteristic of a living thing into another. So this is essentially the main part of this chapter here. You have to have an idea of how genetic engineering occurs. So there's about 5 steps here. If we take a look at this firstly over on the right hand side, you take a gene of interest from a first organism. An example here would be something like insulin production. So people who are unable to produce insulin do not have in some cases don't have the correct gene for producing insulin. So what can be done is you can essentially produce insulin outside of the body. So what you can do is you can take the target gene, it's called, that's the gene that produces the insulin, and then you can take a second organism such as a bacteria. And what's possible to do is you essentially put that target gene into the DNA of the second organism, like the bacteria, and then you combine them together. So it's a combination of the bacteria's DNA and also the gene for insulin as an example. Then what you do is you put that DNA back into the bacteria, and it's called recombinant DNA at this stage. This is where the DNA has been recombined. Second last thing then, what you do is you allow the bacteria to make copies of itself. As you can see here underneath, there's a number of copies of this cell produced. And finally then you'll ask that cell to produce the insulin that you can take out. When you're looking at things like bacteria, they can multiply every 20 minutes, so you can very quickly have millions of copies of a particular gene in almost no time. So I'm just going to pick out the important parts here. Essentially what you need to know is the steps involved in the genetic engineering. And these 6 steps will essentially cover what we just mentioned there on that diagram. So the first one is called isolating. That's when you remove the human DNA containing the target gene from the chromosome and the plasmid DNA from the bacteria. So as mentioned, you take the plasmid out and you take the target gene. Second one then is cutting. The human DNA and the plasmid DNA are cut using the same restriction enzyme. Now a restriction enzyme is basically just something that cuts at a particular sequence. If you ever did the DNA chapter, if a restriction enzyme sees a pattern, maybe something like ATC, it might decide to cut the DNA at that point. And the reason why you'd use the same restriction enzyme is because you want to make sure that the patterns match up. Essentially the idea is you want to make sure that the bacteria doesn't realize that anything strange has happened to its DNA. Now in reality a lot of them do realize and they don't produce the gene that you want, but if you can, I suppose, trick the bacteria into thinking that it's perfectly normal, then you have a better chance of success. Third one is called ligation. This is when the target gene is placed into the plasmid DNA and joins to it. So essentially we've just put the two together. The plasmid DNA may stick to other plasmid DNA and the plasmid DNA may stick to the human DNA. Okay, this term here, DNA ligase, that's kind of like a DNA glue that's going to basically combine them together. Fourth stage is transformation. Transformation is the uptake of DNA into a cell. So what's happened then is now that you've put the recombinant DNA back in to the cell, it says, as I mentioned, only a small number of bacteria take in the plasmid with the target gene. In reality, you only need one to succeed, as one bacteria can make multiple copies of itself. Copies of itself here, this is called cloning. Cloning is when identical copies of a bacteria are produced. And as the bacteria reproduce, they produce copies of the plasmid with the target gene. And finally then, expression is at the end. Now that you've made these copies, this is when the bacteria containing the recombinant DNA produce the desired product. And the desired product must be isolated from the bacteria and then that produced it and then purify it. So you basically have to take the produced insulin out of the bacteria. One of the things they tend to ask on this is, can you give an application of genetic engineering? So you need one for plants, for microorganisms, and for animals. If we look at plants, the first thing we have here is weed killer resistant crops. Some crop plants have a bacterial gene that make them resistant to weed killer. As you might imagine, if you're spraying to kill off weeds, it's very handy if your crops do not die as you're doing this. Another example is golden rice may be a solution to vitamin A deficiency. So there's basically a deficiency of vitamin A in some Asian countries, and what they've done is they've genetically engineered the rice so that it has vitamin A in it, and as a result, the population gets an increased level of vitamin A. Involving animals, there is a certain lung condition that humans have called emphysema, and it's where they're missing a particular protein to basically has to do with their lungs. What you can do is you can get a sheep to produce a protein to treat emphysema. Essentially, the idea is that you genetically engineer the sheep, and then what happens is when they produce milk, their milk will have this particular protein in it. And then someone with emphysema can drink this milk in order to replace the protein that they did not have. And the final example is the one that we looked at the beginning, bacteria making insulin. As mentioned, we can insert the gene for human insulin into bacteria, and then they produce large quantities of this insulin. Of course, you have to consider the ethical issues of this. You have to say, I suppose, firstly here, it says the release of the genetically modified organisms into the environment can be an issue. Sometimes what happens with things that are released into the environment is that they can take over, they can be invasive. Things like you may have heard of things like Japanese knotweed, wasn't genetically modified, but it's a very invasive species that can easily take over an area. There's a few others there. The use of GMOs as a food source is an ethical issue. The fear that animals may suffer, of course, is ethical. And the fear that humans may be genetically modified is another consideration that, in general, these things have to pass ethical approval before they're allowed. So, in summary, although in parts it's a kind of a theoretical chapter, the main things you need to know is the definition at the start. You need to understand the 6 steps involved in genetic engineering, and then you have to have one example of its use for plants, for animals, and for microorganisms. Nice and simple. I hope you found that helpful. What I'll do is I will upload this video, and I'll also put up some questions for you to attempt with that as well. Let me know if there's anything else you need or if it's helpful. Thanks very much.